It's mentioned as 1 p.m. in the calendar too.
Huh?
In the calendar on the website, it's mentioned as 1 p.m.
Yeah, I sent on my seminar list, I said time change at 10:30.
Okay, you're on.
Okay, hello everybody. Thank you for coming, and I apologize for this last minute time change. I tried to send a reminder this morning. At any rate, it's a pleasure to have here Dr. Niethammer from University of North Carolina. He's in the computer science department, and he joined as an assistant professor a couple years ago. He's also affiliated with the Biomedical Research Imaging Center at UNC. He received his PhD from Georgia Tech in electrical engineering, and he was also associated as a researcher at Harvard Medical School, Brigham and Women's Hospital. His research interests are in the biomedical imaging area, and he's doing all sorts of applications related to that. And it's a pleasure to have you here. Please, the floor is yours.
Thanks for inviting me. So today, my main topic will be optimal, fluid registration from an optimal control viewpoint. But because I've never been here, I figured I'll talk a little bit about all sorts of other projects that I have before I go into the meat of optimal control and how it relates to fluid registration. So I think the first 20 minutes, I'll spend probably 1 minute just on some older projects just to fit in context of where I'm coming from. Then application areas that I'm currently working on. And then I'll go towards atlas building and optimal control, which will be related to neuroscience and neurodevelopment. So in terms of where I'm coming from is I've done quite a bit of work on image filtering, image processing, shape analysis, local and global shape descriptors. Also worked quite a bit on diffusion-weighted magnetic resonance images and processing of those. And have done some work in visual tracking and visual feedback control. And just like a 1 minute thing on shape analysis, because this is probably the last main thing I worked on before I came to UNC. So I worked with a colleague of mine on global shape descriptors and spectral shape descriptors. So basically what you do is you take a shape, can be a surface or a volume, and you characterize it by its spectrum, like the vibrational modes. And it's interesting because these things are completely invariant to any kind of isometries. So if you change it, or if you would even fold it, would get the same spectrum, same kind of description. You can use this for statistically comparing shapes or to characterize them in the first place. I'm not going to talk about this in detail, not at all actually. It's the only slide, but if you're interested in talking to this with me afterwards, just let me know. And by the way, feel free to interrupt me at any time if you have questions about anything. In terms of diffusion-weighted images, I've worked pretty much on anything from selecting gradient directions, how you acquire the signal, to processing of the images, fitting models of diffusion, water diffusion in the brain, to more advanced analysis where you try to recover connectivity information in the brain. And I've worked mainly on MR, but recently my interest has more shifted to incorporate other kinds of imaging modalities too. So I work on MR, I work on microscopy images, also work on electron tomography. So you're really looking at very, very different kinds of scales in terms of spatial resolution there, where you're looking at millimeters, you go to micrometer scale, and for electron tomography, where you can have 3D image reconstructions, also you're looking really at the scale of nanometers. So this is an image of vesicles in a synapse in the brain. So from here to here would be 40 nanometers. So very, very high resolution. And what I'm going to do in the next couple of minutes is I'm just going to give you a little bit of a flavor of the kinds of projects I work on with these kinds of modalities that range from segmentation of synaptic vesicles to skin cancer, which is microscopy, to osteoarthritis, which is magnetic resonance again, and then I'll transition to more applications in neuroscience that will be related to the registration method I'm going to talk about later. So in terms of electron tomography, so this nanometer scale, just working with collaborators on methods to automatically extract these vesicles from the 3D electron tomography images. And oftentimes, I mean, if you work with biologists, the interesting part here is that there's a lot of analysis methods out there in like what you would call traditional medical image analysis for segmentation and registration. But if you move toward the more biological problems, it seems like there's really oftentimes a shortage of methods that can actually be applied to those problems and solve the problems for them. So we just came up with like a simple methods to extract these vesicles from the image, and then hopefully ultimately to come up with some sort of measures of the proteins that penetrate the vesicle wall that could tell you then some local characterization of what's going on with the vesicles, if they're different in different parts of the synapse, these kinds of things. And I mean, this is just to illustrate that there are certain tools out there for biologists that attempt to solve the problem, but they're really nowhere at the level of sophistication that you would have in like classical medical image processing, medical image analysis for something like where people study the brain, let's say, or some sort of phenomena at an organ scale.
So this is invasive, right? I mean,
oh, this is totally invasive.
Yeah.
I mean, you have a tissue sample, and I mean, collaborator calls it either like frozen or fried chicken, depending on how you prepare the specimen. It's either like super cold, or you can put this in some sort of how should I say this? I mean, like some sort of you need to prepare the specimen relatively heavily and stain it to be able to see it afterwards. So there's nobody alive anymore at this stage. So, but I mean, to give you a little bit of an impression of the kind of range of projects I work on. Then in terms of skin cancer, this goes one level up in with respect to the resolution. So we're talking about micrometers here. And what we're talking about is really, skin cancer is a relatively, I mean, bad disease to have, because if you don't catch it early, this will likely kill you. And I mean, with sun exposure and all of these things, it's a large fraction of people will get it in their lifetime. And what we're trying to do here is to come up with methods based on histology slides. So these are biopsies. You take a piece of skin, you put it on a slide, you stain it, to come up with methods to classify them into different subtypes, and hopefully in the end also to come up with some sort of classification for or like rating for what the survival chances of the different patients would be. And very far down the road, maybe that could at some point also impact treatment, or how you would what you would do with these patients to increase their chances of survival, give them the best possible treatment. But the challenges here is, one very basic challenge already at the beginning is, if you do these kinds of studies and you do this with, let's say, lots of slides from lots of different laboratories, if you want to push this towards quantitative analysis, then you have the issue that these may not look exactly the same, because people use different amounts of stain, they maybe have different amounts of exposures to sunlight, all of the above. And while you have like methods, for example, in MR imaging to compensate for these kinds of effects, these methods for histology slides are really not very well explored. So with a little bit of effort, I think you can have a pretty big impact there. And we came up with methods to normalize them, bring them into a relatively uniform color space. And then once we have this, we just start with extracting, segmenting nuclei basically, and based on these nuclei, come up with features that somehow characterize different appearances of different types of this skin cancer. And to, like, I mean, in the first step, it would just be a nuclear segmentation. It's challenging because, unlike the classical medical image analysis problems, if you go to biology, those domains are oftentimes very unstructured. So I mean, if you image the brain, everybody has one brain. But if you look at these things, they're like thousands, maybe, or like lots and lots of cell nuclei. So to be able to reliably segment them is actually quite challenging. And because they tend to cluster together very tightly, they merge, sometimes they're not very visible very well. I mean, there's an argument to be made to if it's even important to exactly segment them in the first place, or if consistency is the only thing that matters, if you have a computer analyzing it afterwards anyway. But right now we're more in the exploratory phase, where we came up with a method that segments them relatively reliable, has a post-processing part to it to try to refine the segmentation in terms of like chopping up regions that are obviously wrong. So you see this here a little bit where you have like an area of 3, and you chop it up and you get 3 little ones that presumably are better representations of what nuclei there are in the first place. And then once this exists, we come up with a whole battery of features that describe like the shape of these nuclei, that describe how they're placed with respect to each other, describe texture, and do some form of machine learning basically to try to discriminate them from each other. This being said, this is sort of like a very, very first step at this problem, because if you talk to a dermatologist, they will tell you that a lot of things that matter in looking at them on the slide is not necessarily what they look like locally, but like the large-scale architectural features, where they are in relation to each other, what the nests look like, these kinds of things. And I think this is very interesting for future research, but it's probably also going to be incredibly challenging to do this. But this is where this needs to move in the end. I mean, what we do then, as I said, we use machine learning approaches more or less to classify them based on the features. And what we also found is, I mean, if you work with these collaborators, at the end of the day, if you work in this abstract space, let's say you have 40 dimensions and you get some sort of classification out of it, then it's nice, but you need to go back to the image in the end. You need to show it to the actual dermatologist and make sure that what you found there actually makes sense. I don't know if you have this, you probably have something similar here, but at UNC at Vency, we have this beautiful room. It's called the social computing room, and you can just lay out like these hundreds of slides all around you. It's like a whole room with projectors all around, and that's what we do. So we try to really go from having nice theory to process the images to come up with classifications, but then like close the loop and show it back to the people that actually care about it and make sure that what we find is actually meaningful.
Just looking at the room there, it looks like you've got a whole bunch of papers on the wall, but that's actually projections?
So these things here, these are all sections of slides from different subjects. These are all.
Okay. Are they projected onto those walls,
yeah.
Or are they?
Yeah. So it's a room almost as big as this, and every room, I think there are 12 projectors. They project all around the wall. It's like a big screen. So but it's very nice for something like this, because if you really want to explore the data, at some point it's almost impossible to do this on your computer screen. So if you have something like this, you can really see, is the feature I found, does this make sense? Does it really show up in the whole collection of images you look at? Okay, so I also worked on osteoarthritis. This, I mean, again, goes one level higher now in terms of the resolution. So the resolution is lower, but it goes more towards what I'm going to talk about at the later on if I talk about registration, because this is a problem, again, where has pretty high impact, I think, because lots of people will develop osteoarthritis over their lifetime. Yes?
This is me.
Oh, you have that? Okay. The problem is, I mean, as you probably know, there's no real good cure for this. So the interesting part, though, is that there are databases out there where they acquired a lot of data. And by a lot, I mean, for example, the Osteoarthritis Initiative, I think they followed on the order of 4,000 people longitudinally. So they all have, I think they have X-rays, they have MR, and they have this at different time points, at I think 5 time points if I'm not mistaken. So it's a tremendous amount of data. And the problem with this is that to really analyze this, it's not, I mean, it's pretty much impossible to do this manually, because what matters there is the cartilage that it generates. And if it takes a medical doctor to maybe segment this an hour on just one of these layers, then I mean, you can imagine if you would want to have a large-scale study that analyzes the whole set of these images, it's not feasible, it would take forever. So we're working on methods to try to automate this and hopefully be able to analyze a good chunk of this data set. There's also another one from the Pfizer longitudinal study. It's a little smaller, but it's a little better controlled. But I think these are interesting problems because they have a longitudinal aspect to them, and I'm interested in like registration over time and changes over time. The Pfizer longitudinal study, not yet, but it may be. The OA, yes, you can just sign up. They will, I think you send them a hard disk or they send you one because it's so much data, but it's publicly available, yeah. So the first step we did here is to come up with a way of properly aligning these images automatically. Because while there is, they use certain protocols to make sure that the knee is aligned relatively consistently. So I mean, there are ways where you would just position the patient sort of against the wall like that, for example. It's not perfect. There's always a little bit of variation, and if you're looking for something as small as cartilage, if you want to have a reliable analysis afterwards, it makes sense to try to align them as well as possible from the get-go. And it's an interesting registration problem because it's different than doing brain registration, for example. Brain is just like a little bit of a wobbly mass, but here you really have a joint and you have very different kinds of tissues. You have hard bone and you have flexible tissue around it. So we just were working on methods to automatically extract these bones and come up with ways of aligning it as a first step of this cartilage segmentation problem. And the nice thing I think that comes out of this is that this is really badly visible, but basically
it's better on the screen.
Oh, it's better on the screen, yeah, much better. So you can use methods that I call this. If you've seen active contours before, then what you typically have is you have some sort of term that tries to drive it to an edge, let's say, but it's completely isotropic. It doesn't have any directional information in it. So if you want to encode something like that, you want to get a segmentation that jumps from dark to bright instead of bright to dark, which is meaningful here because on an MR you don't see the cortical bone, it's dark. So if you want to segment the outside of it, you want to go for the transition that goes from dark to bright. If you want to do this, you need to have some sort of way of also incorporating directional information. And people have proposed something called Finsler active contours, which is essentially like a generalization where you have a metric that has orientational dependency. But what you can do is, like with these new methods that are out there that are continuous equivalent of graph cuts, you can also incorporate orientation information. But the interesting part here is, so I've worked on this, interesting part here is that you can, if you set it up like this, you can get globally optimal solutions with this orientational dependency in your surface or curve evolution, which is a very nice property, and we use it for this particular application. So you would just have, just to indicate this, so instead of a graph like you would have it in graph cuts, you solve a differential equation, but then you locally have a metric that is no longer isotropic like a ball, does not need to be like an ellipsoid, like you would have it if you have some sort of Riemannian metric, but can be much more general. And for this image that you've seen on the previous slide with the knee, you would have something that is anisotropic in the sense that it penalizes very heavily in one direction and almost not in the other one. It's almost like a semicircle. But you can put all of this into these frameworks and can still very reliably solve this globally optimal, which is nice, I think. Here are just a couple of results. I'm just going to skip this. We also push this towards like 3-label segmentations and to be able to make sure that we can always discriminate the two bones from each other, and then later if we move towards the cartilage where you have touching objects, that you can still have discrimination between top and bottom and you don't have something that just blurs into each other. So this is sort of a little bit of an overview, and I'm going to move now to this neuroscience, neurodevelopment, which is going to be the meat of the talk in the end. So what I do here is mainly related to, in the end then, neurons in the brain, connectivity in the brain. And I don't know, how many of you are familiar with diffusion-weighted MR imaging, but just to recap a little bit what you can do there in case you've seen it before. So how this works is that if you have a normal MRI, a structural MRI, you get a black and white image out of it, and it will tell you something. There's gray matter, there's white matter, but that's about it. If you do diffusion-weighted MR imaging, you can measure water diffusion in the brain. And the way this works is that instead of one image, you just acquire a whole set of them in different spatial directions, and each spatial direction probes a different direction for the water diffusivity. And you see this here in these images. So this part here is the corpus callosum. In some of these images, it's dark, and in some of these images, it's bright. If it's bright, it means the direction that you probed here, did not have a lot of diffusion. So probably this was like in this direction, up to down, because there's not a lot of diffusion in the corpus. And in other directions where it's dark, it means it diffused a lot in this direction, so probably from left to right. But to make a long story short, if you do this, you can get a reconstruction of diffusion properties in the brain, and you can locally, in the simplest form, like describe this as some sort of diffusion ellipsoid, or you can then take all of this information, paste it together, and come up with these beautiful images that are a macroscopic representation of neuronal connectivity in the brain. Now, the interesting part here is that, of course, you can also do this at all sorts of different scales. So you can do this for humans. You can do this with macaques, and we're actually working a lot with macaques, and this is also what I'm going to talk about later. But even more interesting is that you can, in principle, push this to other modalities and other levels of resolution. So what you see here is a microscopy image, also of neurons. And the nice thing here is if you, while you're working here really on a very macroscopic level, meaning that every voxel probably has millions of neurons going through it, you get a little bit better if you do microscopy, and you can get extremely good resolution, of course, if you do electron tomography. But I think an exciting part that we're starting to work with is the microscopy, because people have mouse models in this particular case that are bred in a certain way, so you can genetically engineer them that only certain parts of neurons light up under the microscope by fluorescence. So you could say, I only want the ones that come from the hippocampus, or I only want the ones that come from the cortex. If you combine this in the end with this, is of course a very powerful method to try to like validate your algorithms, or to have a much more controlled experiment. Okay, so that's sort of the overview. So now let's move to what I really claimed I would talk about in this talk. So talking about registration, optimal control, and longitudinal atlas building. So what I mean by this is, in longitudinal atlas building, what you're after is you have images of, let's say, monkeys in this particular case, or humans, and you have them acquired not only at one time point, but you follow a subject over time. So you acquire it like maybe every year, every half year, I mean, whatever makes sense for the particular application. And what you're after is, or what we are after initially, is to get an idea of what normal brain development would look like. And so, because you can't really necessarily trust one subject, you want to do this for a whole set of them, for a collection of them, and come up with averages, what the average change looks like over time. And this is a pretty challenging problem, and it's a pretty challenging problem for multiple reasons. I mean, one reason is if you would do this on structural MR images over time in neurodevelopment, then you run into a problem that's called contrast inversion. And so just to describe what you see here, in the left column you see a structural MR image at 3 different time points, 2 weeks, 3 months, and 12 months of a macaque. And in the middle column you see, middle column and the right column here, you see images that were obtained from diffusion-weighted MR imaging. So the middle one is what's called fractional anisotropy, tells you something about how directed the diffusion is. If it's very bright, it pretty much means that you're in one of the major fiber bundles. And the one over here is color orientation information. So if it's red, it means diffusion goes from left to right. If it's blue, it goes from the bottom to the top. If it's green, it goes from the back to the front. But the interesting part that you see here is that at very early age, these neuronal tracts, they're already formed. You see them at 2 weeks, right? And while this is pretty noisy data, which also shows you how challenging this is to work with this monkey data, while this is pretty noisy, the structures are pretty consistent already from the beginning. Now, if you look at the structural MR image, it tells a completely different story. And the reason for this is that in neurodevelopment, if you go through for the full range, it takes a while for the neurons to get myelinated. So this is sort of like this cell that just wraps around the neurons. It's essentially like a fat sheet. And if it's not present initially, then even though you probably would want to have something white appearing here, it doesn't appear white because it's just not there in neurodevelopment yet. So if you would want to have an alignment of this image to that image would be rather difficult because it changes appearance. So this is one of the reasons why when we do this currently we are going for the diffusion weighted image, because these structures are just more consistently present already from early on, which will give you a better registration in the end. Okay, so let's talk a little bit about how this can be done. So an approach that exists out there that Brad Davis developed a couple of years back is you just have a couple of brain images, let's say, and they are indexed over time. So it would be young on the left, old on the right. And to come up with some sort of estimate of what a mean image would be, you just pick the time point that you're interested in, and you compute a weighted average. So you have a kernel that weights them over time, and that's just, high at the time point you're selecting, just like a Gaussian or something. And you compute a weighted average of the images. But by weighted average of the images, it's more than just like taking a weighted average of the intensities. It's a weighted average in this warped space, basically. So it takes into account the deformation because you don't want to blur it. Now, if you apply something like this for longitudinal data, then in a way you throw away a little bit of information, right? Because you already went through so much pain to acquire images at multiple time points, you may as well use it. And presumably also it's easier to register within subject than to register across subjects, because at least for humans, it's probably similar for monkeys. Like there's lots of structures, subcortical structures, for example, pretty well preserved. But if you go for the cortex, for example, cortex looks pretty different from person to person. If you move away from like the major sulci, but you go to like the finer details. So presumably, if you have the same subject, you will have an easier time to register over time than if you just mix up more or less randomly. So the proposal here is really just to, instead of doing this weighted average of images, you look at individual subjects independently. You come up with an estimate of how the image changes over time. And once you can do this, once you have this morphing or interpolation defined, you can just warp images to any time point you choose and then compute an average at this already like corrected time point that you're looking at. So you already changed like what the appearance most likely, or like the shape of the brain should be at this particular time point, and then you average at this one. Now, to be able to do this, you need a couple of component algorithms. And the most basic algorithm you need is a registration algorithm. So that registers, that deforms a source image to a target image. And you also need to do this for a collection of images to define this atlas, this mean image. And the other part that you need is a way to infer a time series from a set of images, from a set of time points from an individual subject. So you need basically like the generalization of fitting a line, but generalizing it to the space of image volumes. And what I'm going to talk about is how you can approach these problems from an optimal control point of view, because I think looking at it from this perspective, hopefully I can convince you by the end of this talk, makes the whole derivation of how you solve these problems much easier than what has previously been done.
To be more clear, it's like a generalized interpolation problem?
Basically, yeah. So I think it will become clear in the next couple of slides. But what it is it's like you could see it as registration between this image and that image, but then look at all of them jointly and have something where you warp the image, so you're estimating a velocity field over time, and that's spatially and temporally dependent. And you estimate it in such a way that it tries to match the data points that come at discrete time points as well as possible. Oh, okay. I mean, in terms of previous work that's out there, there's a work by Borzi, Ito, and Kunish that look at optimal control formulation for optical flow. In terms of image-to-image registration, I think the most closely related work that to what I'm going to talk about is the one by Begg, Miller, Trouvé, and Younes that use these I mean, essentially fluid flow registration and come up with a method to solve it for image-to-image case. And then atlas building is the component algorithm and atlas building that I use is pretty much based on Joshi. And the very first approach to approaching this spatiotemporal atlas building problem with this kernel weighting at a time, computing this mean image is work by Brad Davis and colleagues. So, but I'm going to look at this from an optimal control viewpoint now. So to step back a little, if you do image registration, I mean, there are many different ways of tackling this problem, but the two main approaches really are what you would call elastic registration and fluid registration. And the difference between those two methods is that in elastic registration, I mean, in both cases, you obviously want to match the source image as well as possible to the target image. But in elastic registration, the object that you estimate is displacement field, so directly the displacement from one position to the other. And you regularize this somehow to whatever makes sense to you to make your problem well posed. In fluid registration, the problem is slightly different. So what you do there is instead of estimating the displacement all at once, you estimate a velocity field that changes over time. So a flow that if you flow according to this velocity field at unit time, let's say, flows the source image into the target image. So what I'm going to talk about is the fluid registration problem because it fits nicely into this optimal control point of view. So to step back even more, to make this a little bit more basic, what you want to do is you have, if you go back to points, you have a blue point and you want to somehow reach the red point in unit time, and this is space. And what you want to find out is what's the best possible trajectory that brings me from the blue point to the red point. And best possible trajectory means you have some sort of metric that tells you what you consider good. So in a car, it could, for example, be gas consumption. Here, this is related then pretty much to the path length in the end. You want to find the shortest path. If you do this with moving a point, then what you have is a problem that has an objective function, like a control cost, plus some sort of dynamic that you impose. So just x dot equal u, let's say. And if you do this for the image valued case, the whole setup is very, very similar. The only difference is that the equations get more complicated and you move from ODEs to PDEs. That's an optimal control problem. Now, the problem can be formulated in different ways. So you could, for example, say, I want to exactly match my target point. If you do this for this simple setup, you just get a straight line. You could also say, I want to minimize some sort of control effort. So let's say the square of this input integrated over time. And I want to get close to the final state. So it's not necessary to reach it exactly. This is typically what's being done in image registration, right? Because if you have two different images, you may be able to align them pretty well. But chances are you won't be able to align them perfectly because if they come from different subjects, they will, I mean, no matter what you do, you won't be able to have a perfect alignment, or there may be noise, these kinds of things. So you typically settle for free final state. Gets more interesting, of course, if you go to the time series case. Because there you can set up exactly similar kind of problem. You have your system dynamics. You have an energy function that describes which one of the solutions you want to pick, what you consider optimal. The only difference now is that you integrate over a longer time period, wherever your measurement points are, and you penalize, actually you don't penalize closeness to target, you penalize being far from the target, but you encourage closeness to target. So you want to find a trajectory that sort of gets close to all of these points when you estimate this time trajectory. Now, for the first problem, if you move now to the image case, it's a very similar setup. The inexact matching basically becomes a transformed source image. So phi is just a transformation map that you're interested in, compared to the target image, measured in using some sort of metric. You're just L2 distance, intensity distance squared. And the control effort is being measured by the time-dependent velocity field that you're estimating. based on whatever metric you choose here also. But I mean, it's exactly the same setup as before, just now you're not dealing with like scalar quantities, you're just dealing with something that's defined over a whole image or over a whole image volume. And typically, these problems are being set up in a way that you have the velocity here, and you directly have the map that you're after in this part of the equation, which makes it pretty complicated to solve. So if you look, I mean, to solve, I think is the wrong way to say it. It makes it pretty difficult to derive how you should solve it. If you look at these papers, these are like really, really long derivations, and it's quite complicated and easy to make mistakes here. If you look at this from the optimal control point of view, what you say is you just get rid of the map. Who cares about the map? You care about the map in the end, but really what you're interested in is the velocity field. This is, if you have the velocity field, you can reconstruct the map in the end. Because you just, let's say, start with the identity map, flow it according to the velocity field, gives you the map back. Now, the setup then in the optimal control problem is very similar to the original equation. The difference is now you get rid of the map here, and instead of it, you have an image, like the warped source image, according to this velocity field at time point 1. So you have something that you want to optimize, and because you got rid of the map, you now suddenly have system dynamics. So what was the x dot equals u before is an equation that looks like this. So you have a constrained optimization problem. You want to optimize this energy, but under a dynamic constraint. And the dynamic constraint, what this is nothing else than a transport equation. So what this means is if you have this velocity field, it just describes how the image changes if you push it based on this velocity field. So if you've ever done any kind of level set evolutions, for example, it's the same equation because it's also a transport equation. You push the level set around. In my case, I push the whole image around. And then the question arises, how do you solve something like this, an energy with a dynamic constraint? Now, if you had a course on optimization, the thing that immediately comes to mind is you just slap the constraint on with a Lagrangian multiplier. You can do this too here. The difference then is that it gets a little bit more complicated than what you would see in a typical optimization textbook, because you don't have one Lagrangian multiplier. You have a Lagrangian multiplier function that depends on space and on time. But if you do this, you get a structure that looks like this, which I think is really interesting for the following reasons. So what you get is, you get your initial state dynamics back, just what you stuck in, right? The constraint you get back, you always get this back. Then, though, you get a dynamic equation for the adjoint, for the Lagrangian multiplier. The dynamic equation for the Lagrangian multiplier does not have an initial state. So for the image here, the initial state is just the image I start out with, my source image. For the adjoint, I have a final condition that is related to the mismatch between the target image I want to match and the warped source image. So, and my final condition is based on how well I aligned those two images. And then what the adjoint equation, the Lagrangian multiplier, needs to fulfill is this particular equation. And this particular equation, what this is, it's a scalar conservation law. And the scalar conservation law tells you basically is you, at time point 1, you have this error in the mismatch. If you distribute this backward in time, you'll conserve the amount of error. So if you have compression, it will go up. If you have something that dilates, it will go down. So this is what this, so error is being preserved in this equation. And then the last equation that you have is a compatibility condition that ties all of these conditions together. The velocity field that you're after, the image at all the time points, and the Lagrangian multiplier or the adjoint. And if you have the optimal solution to some sort of magical procedure, then all of these conditions need to be fulfilled all over space and time. Now, the question is, how do you get this solution? And it turns out that if you set it up this way, then the solution method immediately pops out too, because it's an adjoint method that you can use to solve this. And the way this works is goes in the following. Now, you assume somebody gives you a velocity field, an initial guess. So if you don't know anything, you can just assume this is 0 everywhere initially. So it just won't move at all. So you have this, and you start with the source image that you know. So you take the source image, you apply the transport equation, and you flow it forward in time based on the current estimate of the velocity field you have. And you just store it at all the time points that you use to discretize your time, because you need to discretize time. Then once you have this, you can estimate, you can compute what the warped image at time point 1 is. If you do this, this immediately gives you the final condition for the adjoint, because it's just the difference between the target and the warped one, which you just computed. Once you have this, you can just, you know what the adjoint needs to fulfill, because it needs to fulfill this scalar conservation law. So you flow it backward in time, and you save all the values for the adjoints according to this equation at the time points you chose. Now, if you would have the optimal solution, then you could apply this compatibility condition and use at every time step information about the velocity field, the image, and its adjoint, and evaluating this expression would need to give you 0. If you would have this, then you would have the optimal solution. Chances are you don't have this because you iterate to the solution. But then the interesting part is if you go through the equation, then this is the optimality condition. But if you're not at the optimum, then this left-hand side is nothing else than the gradient of this energy with respect to the velocity. So you can use this to, this computes the gradient, and you can use this to locally adjust what the velocity should be at a particular time point, and just iterate through this process to convergence, and that will give you a registration method. Now, what am I doing for time? Okay. So once you have this, once you know how to register 2 images, you can push this one level farther to the atlas building problem. So instead of just registering 2 images, you would have a whole set of images, and you want to simultaneously register them to a mean image that you also jointly estimate. But it's the same kind of component algorithm. It's almost the same equations if you would write it down, just that you sum over all of them and you register to this unknown mean image. And what's being optimized there is really, it's the sum over the square distances of all the images to the mean image. And the distance is being measured based on the velocity field that you estimate, because that gives you a measure for distance, how far you traveled. Now you can do this. If you do this, then essentially you have one of these component algorithms that you need to build up this longitudinal atlas building method. Because
so how did you get your mean shape, your mean?
Oh, this mean shape.
Yeah.
You estimate this jointly. So initially you would, let's say, Sarah and Joshi pretty much, or other people developed this method. I'm just applying this algorithm to it too. But basically what you do is you have an initial alignment, let's say affine of these images, you average them, and then you, it's sort of like jointly registering the images to the current mean. But while you do this, the mean itself changes, and eventually it will converge to the mean image, and you'll get all these velocity fields from all the images jointly. So it's a joint optimization problem in the end.
This is done on a cross-section basis or on the whole brain basis?
Whole volume. Whole 3D volume.
It's not done cross-section by cross. I'm not comparing this cross-section with this cross-section with this cross-section, but you're using all 3D
yes.
at one time.
Yeah. So it's a 4D problem in a way, because you look at 3D plus time. So if you have this mean image, then the only thing that's left that you need to do is you need to come up with a way of the last piece that's missing in the algorithm is to be able to solve this longitudinal registration problem. Because remember, what we want to do is we want to warp images to a specific time point and then apply. I'm just showing 2 here, but you can have say 10, 100, however many you want to have. Warp them to a time point and then use this atlas building method from the previous slide on it to compute what the mean for the specific time point should be. So to address this problem with multiple images, you can use the same kind of optimal control setup. The only difference then is that, again, you penalize the regularity of your velocity field, or the irregularity of your velocity field, And then you have this term that tells you how far my warped image deviates from a measurement at a certain time point. But you just sum over all the deviations at all the time points. And again, you solve it. And the nice thing about this here is that if you go through the derivations are very, very similar. The only difference is that they get slightly more complicated, and what you get is that you get what you call jump conditions for the adjoint. So what this means really is that the continuous dynamics in the forward direction is exactly the same. The compatibility condition that you use to compute the gradient is exactly the same also. And the backward direction, where you have the scalar conservation law for the adjoint, is also the same, but only within intervals of where you're living sort of like between two images. So you would start at your final time point, get the final difference between what the image should be and what the image is. Then you flow this backward, and if you see a new measurement, then you have jump conditions that change the state of the adjoint and push it in a direction that's meaningful to optimize this overall optimization problem. Then you keep on flowing again, and in this interval the scalar conservation law holds, and then you jump again and keeps on going like this. But the overall principle is the same. Then you would flow forward, flow backward, compute the gradient, update the velocity field, run to convergence.
So the underlying assumption here is whatever measurement you made of these images, it's all under the same conditions and everything else. Everything is controlled, in other words, right?
I mean, the better controlled it is, the better, yeah.
Yeah, but that's the assumption, right?
That's the assumption. Yeah. I mean, if you have something that like is not very well controlled, and suddenly you would have a brain that looks very dissimilar. I mean, we'll still capture it, but at this stage, then it's probably not very. Will be like an outlier basically in your data analysis.
Well, I mean, what I'm thinking of is, how well do you cope with variations? Because these things are done at distinct points in time, and any perturbation basically can ruin your life, right?
I mean, I wouldn't formulate that as drastically, but I mean, any strong perturbation is not good for your analysis. But that's why we're doing monkey studies where you can control this relatively strictly, and then if you go towards the atlas, then if you have multiple trajectories, then you do averages at time points, but you do it over, let's say, 20 animals or something like this. So you would get an estimate of the mean image, but also would estimate some sort of local variation of what you expect to see. So then if you have something that really went very wrong, then you should hopefully be able to pick it up at this stage. Okay, so now once you have this, you can compute the image at any time point, because you can just you start with the source, you estimate the time-dependent velocity field, so you can flow it to wherever you want to flow it to. Any point in time is fine. You can reconstruct what it's supposed to look like at this time point. And then once you have this combined with this cross-sectional atlas builder, you essentially have all the ingredients to compute this simple version of a longitudinal atlas. And I mean, here's a synthetic example where these are three images. We just warped them synthetically, and then we reconstructed. I mean, I did this time series registration, and it sort of like matches them visually pretty well. But what you see here is really this like gradual evolution, how the shape changes and matches these images in between. So if you pick any time point, if you decide you want to know what the likely shape is here, you can just pick it at this time point and it'll get some interpolated image. It's just like morphing. Okay, so now we have all the ingredients to do this, and basically then, as I've shown you before, I mean, what we want to do this is with diffusion-weighted images, just because they're not prone to this contrast inversion issue. So you could potentially go back really far in the development, start with really young babies or really young monkeys and do this. But the overall principle is the same. We just use like FA images at different time points, register them to each other, for these individual subjects, get these time trajectories, and then build at whatever time point we choose a cross-sectional atlas that gives you a mean shape at a particular time point. And I mean, just to step you through the process, so you would have these two time courses. You somehow select this is the time point you want, compute the mean image, and then to do any meaningful analysis, also in terms of statistics, you should bring all of this in this coordinate frame. And because you have all these maps, you can easily do this. So you can just decide if you know how this one warps to here, and you know this from the time series, from this registration of those three images, and you know what the map is from here to here, then you can just compose them, and and you know what the map is from here to here. And then you can do this for all of them. You just pull all the information, diffusion information if you like. You represent all of this in this warped coordinate system, and the second you are at this stage, you can use statistical longitudinal modeling, let's say, that takes into account like the temporal changes, the changes over time within subject, but also that you have multiple subjects, and you can come up with nice statistical methods of estimating what the signal should be. Yeah, and you just end up with this. And okay, it's again better visible here. So this is, I mean, this is all. I mean, I apologize that I didn't really bring like my completely finished work. So it's all a little bit work in progress, fresh off the press. But so this is just an example of one of the time points where we averaged, I think, 7 monkeys going through this full pipeline. And so we'll do this at all the time points now, and then you'll be able to see how it really evolves over time. But I mean, the important part is if you use these kinds of methods, you get sort of, the noise gets reduced because you average, but because you use this deformable registration, it's not like you blur the hell out of the image. I mean, you still get all these anatomical details that make sense, and you get a relatively good image. So, I mean, if you remember, I don't know, like 10, 15 slides back when I showed you these monkey images at different time points, 2 weeks, 3 months, 12 months, you can probably appreciate that this is a much smoother, much cleaner looking image than it was before, because this data is really relatively low resolution and relatively noisy, so it's not that easy to work with. I mean, the last thing I want to talk about here then in this context is that there's always also a little bit of a interest in designing algorithms that have certain symmetry properties for registration. And what I mean by this is that if you take your standard off-the-shelf registration algorithm and you register image A to B, and then you try to register image B to A, well, chances are it's going to give you different results because it's not set up in a symmetric way. So what I mean by this is that typically, if you look at the left image, I mean, you dedicate one image as the source, and then you warp it to the target. Now, because this is not symmetrically set up, you get these different results if you would run it in the other way. So people have looked at this problem and tried to come up with ways of symmetrizing this problem. And I mean, the easiest way is just to use some sort of like meet in the middle strategy. So this is like image 1, image 2, and you just register it to meet in the middle to make them as close as possible. You can do this. You can also do something where you just do it continuously. So you warp the target, like image 2 to image 1 and image 1 to image 2, and you compare at each time point in between, set up a cost like this. This will also give you something that's symmetric. The problem with this is that, like this midpoint rule, for example, does not really translate to more than 2 time points, because if you have 3, if you have 4, if you have 5, what's the midpoint? I mean, I don't really know. I mean, you can put something in the middle, but it's not in the middle. It's not symmetric with respect to all of the points. This one you could probably extend, but it gets very costly because you need to do comparison at every time point of your time discretization. So the simple solution, what we did is just to say, instead of having a fixed initial point and a free endpoint, you can just leave both of them free. So you have free initial condition, free final condition. And the nice thing if you do this is that this then completely translates to time series, because there you would also just leave the initial condition and the final condition free, and you have all these. It's not asymmetric anymore at this stage. And what you do then is you estimate the initial template, because it needs to start at a certain value, plus the flow field that you're interested in. And just as a toy example, what this looks like is something like this. So let's say this is the source, and this is the target. So if you do this joint estimation that is symmetric, then what you would get is that you get the deformation field, sort of like that warps this one to that one. So this is like the warp of the coordinate system. But you also get at the same time the template that is some sort of like compromise between the two of them. So where both of them are black, the template will be black. Where both of them are white, the template will be white. In the middle, you get something that's sort of a little bit grayish. So because it's a template that tries to be equally far away from both images to make everything symmetric. And of course, I mean, what I'm sweeping a little bit under the carpet here is if you would truly want to have something that exactly gives you the same result if you run it in both directions, then you would get into the issues that your numerics themselves would need to be perfectly symmetric, and also the area where you represent the solution in which domain. If you go for something big to something small, or for something small to something big would matter. So I'm not talking about this here. I'm just talking about symmetric in theory. And then, I mean, as I said, you can do this for time series. Then again you would just have a free initial condition and a free final condition, and you would have something that is symmetric. And if you go through all these derivations, you end up with sort of like ugly-looking equation. But what it basically means is if you're having a given velocity field and you want to compute the associated template with it, then it's like weighted averaging in whatever reference frame you choose. But the weighting is not just like taking a mean of the intensity values, but it needs to be weighted based on the amount of transformation this an image underwent. So if you're representing everything here, then if you pull this to here, if you pull that to here, then the amount of deformation needs to be incorporated into the weighting scheme to really estimate this properly. And I mean, here's just an example where it's like iterating this template. So it was 3 iterations in the end after running this, and after the second one, it's essentially converged. And what you see is it gives you, I mean, for this specific case, I think I had 5 images in the time series. You can compute this. It gets a little blurrier because you're averaging, but at least like the essential structures are still being preserved. So you see pretty crisp ventricles, and you see some of these white matter structures that are still there. So you can compute this in practice. It's pretty much all I had for this talk. I mean, I think what I try to do here is sort of like lay down the basic principles of how you can approach this with optimal control. But I think for future work, it's the more interesting part comes into play then if you move this to different kind of dynamic models, because the dynamic model that's currently being used is, in a way, is highly trivial, right? It's just this transport equation, you push it around. But I mean, there's nothing that prevents you from having dynamic models that are higher order. So you could think about having something like a spline in image space, for example. Or you could have dynamics that describe mass preservation. I mean, this actually, if you do this, you get something that almost looks like the registration method I had. Just the state equation and the adjoint equation, they switch. So the state equation is scalar conservation law, and the adjoint equation becomes a transport equation. But I mean, I guess what I want to say is you can put in different dynamics. You could, for example, set up a registration method that automatically is mass preserving in exactly the same way with the same kind of math. And you can think about all of like pushing this to like image-based interpolation, approximation, filtering methods. I mean, this is just the beginning, but I think there's a lot of potential to push to more interesting structures in the future. It's pretty much all I had. Any questions?
Questions? I have a question.
Yes.
Pertaining to, we actually had the same, as I was briefly talking to you, we worked on the space of curves, right?
Yeah.
And where we're trying to capture these trajectories and things like that. And now I think the problem basically it's like trying to make your system Hermitian, right? In a sense, you want to run it forward and backwards, right?
Yeah, in this particular case, yes. Yeah.
Right, but fundamentally, I mean, fundamentally there is a problem, right? I mean, physically.
I think you're right in general, but I mean the models that are being estimated here, there are these like diffeomorphic maps. So you don't assume that you have like any kind of like shocks or
Right.
weird effects going on.
Right.
If you would assume that you have this, then I mean you're right. I mean you can't really run it forward and backward. But if your assumption is that you have, due to this regularization, a well-defined map, then I think it becomes meaningful to do it backward and forward. Otherwise, maybe not. Yes.
And the other question I had was more qualitative in the sense where why are they interested in this exactly? Getting all these maps for.
Well, so they're interested in it in the first place because I mean there is weirdly enough apparently it's not completely clear how the brain really develops. So for this particular application, it's really just coming up with a method that would give you a reliable estimate of what a mean brain development trajectory would look like, potentially with some sort of variance estimates on top of it. And then, I mean, you could think of if you have any kind of population that has a specific pathology, you could just also find out what, let's say, the average brain and the variance looks at a particular time point. You compare it across the normal atlas, and you see where the differences are. So in a nutshell, I mean, this is the idea in the end.
Are the times you're working with then, I tend to think about these things as looking at MRIs there, but are the times you're looking at now talking about years of following development?
So for humans, it would need to be years. For the monkey, what we're looking at is, I think, from age 0 to 4. And the reason for this is that I think a 4-year-old monkey corresponds roughly to a 30-year-old human, because they develop much quicker.
Really
so it's
for
a large
long
time
area
span, like a
growth and so
really
forth
full
and
development trajectory for a brain. I'm not talking about like limited points here.
Okay, but that same thing goes on to any other kind of developmental process, the maxillofacial area type things where they're doing cleft palates and the like. Okay.
Yeah, it's much more general. I mean, this is just the driving problem in this particular case.
And there's one more qualitative question. I think there was one slide where you showed where there the mean images, right? So the mean image in this case would actually look like a brain, right? If the doctor looks at the mean image, he would actually think that it's maybe a diffusion weighted MRI, not a fabricated image.
The doctor would probably think it looks like a pretty noise-free MRI. Yeah.
Okay.
Probably would not look. It would look unreal in the sense that it would look too good to be true.
Okay. But it does look like a brain.
Yes, it does look like a brain.
All right, let's thank our speaker. So fundamentally, I think the problem is you do have in the development area, you do have topological changes along the way, right?